Good morning, good afternoon, good evening, everybody, and welcome to our short seminar series. It's our pleasure to host today 4 excellent scientists. And if you'd like to participate yourself, share your research with the TPD community, you're all invited to join our next call for the seminars. And that would be on September 29th. The abstract submission for the next call is August 4th. So please keep that time in mind. You can also find it on our website. So for the webinar today, please use the Q&A panel to ask any questions you may have. And in the chat, you will find a Zoom link to the breakout room discussion where we will disperse after the talk. So that's a great chance to meet the speakers, ask them any follow-up questions, and have a discussion. So with that being said, it's my pleasure to introduce our first speaker of today, Helen Yu. Helen is currently a visiting scholar at the Stanford AI lab, and Helen has started out as a synthetic chemist and became interested in identifying biochemical pathways that explain how small molecules worked. In her graduate studies, she worked on characterizing imide compounds and then became fascinated by data science. After graduating, Helen became interested in programming and she moved to Don Droer's lab at Stanford. Her current work focuses on using artificial intelligence to design new drugs. Welcome, Helen, and the floor is yours. Thank you.
Hi, I'm Helen, and I am a software developer in Ron Droer's lab at Stanford University. And today I'll be telling you about the work that I've been doing in his lab, training software how to draw molecules. And so one of the things I've been really kind of motivated by is that protein structures are much more accessible than ever due to protein structure prediction software like AlphaFold. But on the flip side, our ability to predict ligands that go into those structures hasn't really scaled proportionately. And so my work primarily focuses on this lead discovery task, where you start out with a fragment of your ligand of interest, and you expand it out into a much larger, more lead-like molecule. And so one of the ways that a lot of people solve this problem currently is that they do a brute force search through all of chemical space with traditional docking type methods. And so these methods tend to have around a 5 to 52% hit rate. And one of the problems with these methods is that you really need a really premium set of eyeballs to manually go through and sort through all the dock structures, because a lot of the top physics-based scoring algorithms don't account for solvation because of how computationally expensive it is. And so the result is that, this creates a bottleneck in this process, because as you can imagine, generating 1,000 times more eyeballs is going to be really difficult. And so a lot of my work focuses on instead trying to predict the location in chemical space that your desired compound is going to reside in. And so I thought this was a really similar problem to something I was really interested in as a little kid. And so just the way that artists simplify space by decomposing it into lines, we thought maybe you could simplify chemical space by grouping it together into common functional groups. And so what I did was I wrote a base software that started out with a fragment of a ligand inside of a protein pocket. And you could determine where you can add an additional fragments by looking at the places that had a free hydrogen and replacing it with your fragment of interest. So like a methyl group or a carboxyl group. And so I was really happy that, this worked. But on the other hand, I was really sad because, the quality of a lot of the compounds I generated with this first version of the software was basically fan art quality. And so this example really highlights why this is such a challenging problem, and perhaps why you need machine learning to solve this problem, because there's multiple different places where you can connect atoms. There's multiple different types of fragments that can be connected to your starting ligand of interest. And finally, there's multiple different ways that your fragment can be oriented to your starting template. And so instead of ending up in the place in chemical space that you wanted, you end up in ending up in all these other places in chemical space that would probably make your advisor really sad. So one of the things I was curious about is, can you train software to put the correct fragment, the correct place, kind of the way an artist learns how to draw. And so if you're an novice artist, you look and you see where does an expert cartoonist attach a line and what line it adds at a certain position. And so just the same way, if I start out with a fragment of a ligand, I would train my software by basically teaching it, where do ligands in the protein database, like what are the positions what is the position that an expert ligand would have the next fragment attached to, and what fragment is usually attached to that position. So we use a two model system in order to get this to work. And so first, we start out with a fragment of a ligand inside of a protein pocket. And what we do is we look at all the places where you can connect the next fragment by looking at the hydrogens. And we use one model that we trained to score all those positions relative to each other. And we pick the position with the best score. The second thing we do is, once we have a position, we enumerate through all the different possibilities of different fragments added at different dihedral angles. And we use a different second model to rank all of those possibilities relative to each other. And we repeat this process until we reach a predetermined molecular weight threshold. And so one of the things that we were really heartened by is that if you compare the molecules generated with our AI software, our trained AI models, and compare it to molecules generated with the standard physics-based software, you can see that the molecules that our AI software generates actually do a much better job of fitting into the protein pockets. The most egregious case, of course, would be this salmeritol example. The second thing we did is that we benchmarked our compounds that our AI-guided molecule generated across a number of different physicochemical properties, and compared it to ligands in the protein database, which is in the green, as well as proteins generated with the standard physics-based algorithm, which is in the blue. And so I hope you can see across all these different properties that the red line, which is our AI-guided molecules, does a much better job of overlapping the green line, which is the reference drug-like molecules, and the blue line does. And there's two things in particular that I would like you guys to pay attention to. And so those are the panels on the bottom left. So that's a number of unsatisfied hydrogen bond donors, and the panel on the top right, which is the amount of formal charge. And so as you can see, the molecules generated with the standard physics-based algorithm does a really bad job of recapitulating the distributions of drug-like molecules in the protein database. And I think this actually shows why you need like a premium set of eyeballs to go through and sort through the doc structures. Because this software is not going to account for solvation. And the other thing is that how much formal charge and how many unsatisfied hydrogen bond donors the pocket can tolerate is going to be really dependent on the pocket of interest. And you really need someone who's had a lot of experience looking at protein ligand structures and understands the system to figure out where those thresholds are. And so the next thing we wanted to do is to figure out exactly how interpretable were these models that we trained. And so the first thing that we did is that we did an experiment where we took a methyl group that was attached to a template ligand and rotated it around 120 degrees and just looked at like what score our model gave out. And so as you can see, a compound with the staggered conformation has a much lower score with our model than a compound with an eclipse conformation. And in fact, you get something that looks very similar to like a potential energy function, which we were really excited by. The second thing that we did is to look to see if our model was sensitive to different amino acids in the protein pocket. And so in this experiment, what we did is that normally the top ranked fragment added is a carboxylate to make an interaction with an arginine in the protein pocket. So what we did is we mutagenized that arginine into a leucine. And as you can see, the top ranked fragment ends up becoming this hydrophobic group instead. Finally, we wanted to see whether or not, the model that we trained can make inferences about fragments that it's never seen before when we do the training. And so what we did is that we had a native ligand with the pyridazole fragments that our model has never seen before, and we give it an input structure with that pyridazole fragment removed. We then add that fragment back in to our library, and we generate and score around 500 different candidates. And what we saw was that a compound with a pyridazole fragment was actually the second, highest scoring compound, which we were really excited by. Two ways that this software can be used in the targeted protein degradation space. The first is if you're interested in generating a new scaffold against your favorite E3 ligase or deubiquinase, then you can use the software to derive novel scaffolds if you have a crystal structure of your ligands in complex of your protein of interest. And the same goes for targets that you're interested in. For example, if RAS is a protein that you're interested in discovering new drugs for, then you can just use the crystal structure of the RAS G12C mutant, bound to AMG 510 in order to generate a new scaffold. So there's three main points that I hope everybody takes away from this talk. First, we desperately need to figure out how to discover drugs in a way that's a lot more high throughput because of protein structure prediction software like AlphaFold, and having to go through and manually pick out hits because to compensate for methodological weaknesses in most docking software creates a pretty big bottleneck. And the software that we develop can potentially open up this bottleneck. And so speaking of bottlenecks created by sudden surges in demand from industry, Ron is always looking for independent and courageous students who are interested in pushing the forefronts of drug discovery. And this work would have never been possible without, without the two very talented graduate students I was very fortunate to be able to collaborate with. Alex Powers, who is a student in the chemistry department, who has developed a lot of really great educational software. And Patricia Suriana, who is a graduate student in the computer science department, who had worked as a professional software developer before coming to grad school. And thank you for all your time.
I'm very happy to introduce Lena Nitsch. Lena joined us for the internship at the Broad Institute in the Ebert lab, where she focused on the mechanisms of the drug-induced BCL6 polymerization. Subsequently, it was my greatest pleasure to supervise Lena during her master thesis in the laboratories of Professor Dr. Claudia Scholl and Professor Dr. Stefan Fruhling at the DKFZ, where she used a minimal BCL6 domain for reversible drug-induced oligomerization as a molecular switch. Currently, Lena Nitsch is a PhD student at the Ludwig Lab at the Berlin Institute of Health in Germany.
and thank you so much for giving me the opportunity to talk about a project that is very close to my heart and that involves the BCL6 BTB dimer and how it can be used as a novel building block for drug-induced reversible oligomerization. This project actually started off as a side project from a very exciting study that was published in 2020 by Nikolai Slavicki, Ho Jung-yoon, and Jonas Kappel, focusing on the mechanism of BCL6 degradation induced by a small molecule. And to give you a little bit more background on this, I have here a very simple schematic of BCL6 dimer. So BCL6 is only stable in the stemeric form. And important for this dimerization is the BTB domain that you see here and also down here. So here you have two BTB monomers that form a homodimer, and within this dimer you have a dimerization groove, and within this groove this particular small molecule binds, and this small molecule is called BI3802. BI3802 was partially described in 2017 by Böhringer Ingelheim and was then made publicly available via the OpenE platform. And what BI3802 does is that it binds to the BTB domain of BCL6 very specifically, and then oligomerize BCL6, what you see also here in cells. And upon oligomerization, BCL6 is then degraded. Important for degradation here is CR1, which is an E3 ligase that has a binding site in BCL6 and was shown to co-localize with the BCL6 foci in cells. And what happens here in more detail in vitro is that BCL6 forms upon addition of BI3802 those fibers that you see here in this electron microscopy image. And that all led to this model that you see here on the left-hand side, containing the BTB domains that are then kind of connected with BI3802. And in vitro, this fiber formation is actually dependent on compound concentration. So we have here a low concentration versus a higher concentration, and you see very prominently already in these images, but also quantified here, is that lower concentrations lead to much shorter fibers and also less dimers that are present compared to higher concentrations that lead to much longer fibers, as well as more dimers involved, suggesting that in vitro oligomerization here is dependent on compound concentration. In addition to this, we also saw using DKFZ that the oligomerization is dependent on treatment duration. And so you see here, shorter treatment time also led to less TFZ signal, and the TFZ signal seemed to be saturated at around 60 minutes of treatment time, suggesting that this could be a very interesting tool to use this to study oligomerization. But for that, we also wanted to see if this works in cells. But for this, we first had to overcome this obstacle of having a CR1 binding site, because that would ultimately always lead to degradation of a construct. So we designed a minimal construct that just contained the BTB domain and fused this to GFP for easier visualization. And we saw that this construct is, upon BI3802 addition, very stable and it's not degraded compared to longer constructs. It still forms very nice foci that are actually stable over multiple days. And another thing that I didn't tell you yet is that there's also a second compound that is actually very similar to BI3802, so this oligomerizing compound, which is called BI3812. It just differs in this one particular part here that is also colored differently, and because of that, BI3812 is not able to oligomerize BCL6. It still can bind into this dimerization group though, because it's so similar. And how we use this is that we can actually add excess of BI3812 to revert the oligomerization and foci formation induced by BI3802. So now we have an oligomerizing compound and a de-oligomerizing compound. And this actually is not only possible in one cycle, but actually in multiple cycles, which is very exciting because now we have a very tunable system also for protein oligomerization in cells. And then we brainstormed of how we could use this. And the first thing that came to our mind was receptor biology, because we knew that a lot of receptors were active only upon oligomerization. So we thought if we just introduce a BTB domain in a receptor of our choice and then express this in cells and then treat those cells with BI3802, we could potentially activate the receptor by oligomerization. And then if we just add excess of BI3812, it would compete for the binding site, and then we could quite rapidly stop activation by stopping the oligomerization of our receptor. And that's actually what we did. So we chose a very well-studied receptor system, which is the EGF receptor, and introduced the BTB domain intracellularly of the receptor. And because it's so well studied, we knew that the EGF receptor is only active or fully active upon oligomerization, and that you can assess the activation status quite easily by checking the phosphorylation of the receptor itself. So if it's phosphorylated, it's active, and similarly also the downstream signaling molecules, predominantly MEG, ERG, and AKT. So if they are phosphorylated, it suggests that they are active. And then we took our fusion receptor and expressed it in two different cell lines, ecto-9 T cells that are not EGF dependent, and MCF10A cells that are very dependent on EGF signaling, and treated those cells then with BI3802 and looked at the phosphorylation status of the receptor and the downstream signaling molecules. And what we observed here compared to the parental cell line is that upon expression of our fusion construct, we were able to increase phosphorylation of all downstream signaling molecules as well as of the receptor itself, suggesting that we did induce some sort of activation of our fusion receptor. And to then have an additional physiological readout, we only turned to MCF10A cells, because here you actually supplement EGF to the normal growth medium for them to proliferate. So we performed a colony formation assay and to check the cell growth. So we seeded cells at low density, and then either did not supplement anything as a negative control, and you see nearly no cell growth, or we supplemented EGF as a positive control, where we see an immense cell growth. And then what excites us the most is that if we don't add EGF and just supplement BI3802, we don't see any cell growth in the parental line, but we do see cell growth at a comparable level to the EGF addition here, suggesting that our receptor is actually physiologically relevant in the cell lines, leading to proliferation, which is very exciting. And lastly, we also checked if we can revert this protein activation by addition of BI3812, so this de-oligomerizing construct, and here we only check the phosphorylation status of the receptor itself, because this is the most prompt response. And again, if we only add BI3802, we again saw a strong phosphorylation compared to the DMSO control. And then if we add BI3812, we nearly diminish the phosphorylation of EGFR BTB, which suggests that we at least partly revert the protein activation, which was also very exciting. And with that, at the end, I would just like to sum up by telling you that we found a novel chemical and synthetic biological tool that's quite precise, because BI3802 and 12 binds specifically to the BTB domain of BCL6. It's very regulatable, at least in vitro. This is dependent on compound concentration and treatment duration, and it's reversible because we have BI3802 as an oligomerizing tool and BI3812 as a de-oligomerizing tool, with which we were able to investigate functional consequences of protein oligomerization in the EGFR setting, but also we anticipate that this will be possible for any protein of your choice. And if you're interested in learning more about this, we also were quite happy that we were able to recently publish the study in our reports methods. And with that, I'm at the end, and I would like to thank the whole team that was involved, in particular my master thesis lab, led by Claudia Jeff Frühling at the DKFZ in Heidelberg, Ebert and Fischer labs at the Dana-Farber, in particular Ho Jung and Shuya for the in vitro work. And of course, the biggest thank you goes to Mikai, who's not only the most brilliant and most enthusiastic scientist I've ever met, but was also the most caring and amazing mentor, without whom this project would have never happened. And I would just like to take the opportunity to thank them for everything. And lastly, I would also like to thank you all for your attention.
It's my great pleasure to introduce Natalia Szulc. Natalia is a second-year PhD student in the group of Wojciech Pokrzywa at the IIBCB in Warsaw, Poland. Natalia obtained her master's degree in molecular biotechnology and computational engineering at the University of Warsaw. Her PhD project focuses on the evolutionary adaptation of the proteome to regulation by the ubiquitin proteasome system. Her main research includes the mechanisms used by proteins to avoid premature degradation, degron evolution, and substrate specificity of ubiquitin ligases, which she dissects using machine learning and structural bioinformatics. She recently received a 3-year Pluridium research grant for her PhD project from the National Science Center in Poland. Natalia, welcome.
Hello, my name is Natalia Szulc, and I am a PhD student in Wojtek Pokrzywa's group at the International Institute of Molecular and Cell Biology in Warsaw. It is my pleasure to present our new web server, Degronopedia, which allows for proteome-wide inspection of degrons. The ubiquitin proteasome system is a principal proteolytic component of the cellular proteostasis network. It involves an enzymatic cascade which mediates protein ubiquitination and degradation. One class of enzymes operating within the ubiquitin proteasome system are E3 ubiquitin ligases, which recognize substrates through their short linear motifs termed degrons. Upon substrate binding, the E3 ligase mediates the ubiquitin transfer onto the substrate, which often leads to its proteasome-dependent degradation. And I wanted to tell you here that our idea to develop Degronpedia actually arose from one of our projects where we needed to screen several proteins for the presence of known degron motifs, yet the sparsity of bioinformatics degron-dedicated tools made this task very challenging. And for this reason, I developed Degronpedia, which provides detailed information about degron motifs occurring in the query protein. Degronpedia accepts 3 types of inputs. It can be either a UniProt ID from selected model organisms, a protein sequence, or a protein structure. Then the server performs the degron calculations, and as the output, provides all occurring degron motifs in the query protein, as well as lots of other data. And let me now tell you how Degronpedia works on the example when queried by UniProt ID, because this query type provides you with the most comprehensive degron output information. So I collected data from 7 model organisms, and by this data, I mean their complete reference proteomes and their corresponding alpha-fold structures, data on N-C-terminus stability, data on post-translational modifications, E3 substrate, interactons, pathogenic mutations, or experimental cleavage sites, and deposited them in a dedicated database. So when the server receives the query, it screens the protein sequence for the presence of known degron motifs, and next it presents them in the tripartite degron architecture context. The degron tripartite architecture was postulated by Guher and colleagues, and it assumes that 3 components need to be present so the degron actually acts as degradation targeting. And these components are the primary degron, which is the very motif recognized by the E3 ubiquitin ligase, the secondary degron, which is located in the neighborhood of the primary degron, and this is a residue onto which the ubiquitin transfer may occur. And I wanted to emphasize that in our implementation, these are not only lysines, but can also be cysteines, threonines, or serines, because these residues were shown that may undergo the so-called non-canonical ubiquitination. And finally, the tertiary degron, this is intrinsically disordered region in close proximity to the secondary degron, which acts as an unfolding seed, initiating proteasome-dependent degradation. So for each found degron motif, the Degronpedia reports all their potential secondary and tertiary degrons. So finally, I overlay all the degron motifs with the collected data I have shown you on the previous slide, and I provide the comprehensive output in the form of tables, as well as visualization on the sequence. And before I show you the Degronpedia in action, I just wanted to tell you about one of server's features. So Degronpedia allows the user to simulate the sequence cleavage by a number of different proteases, and then it screens for the degron motifs in the newly emerged terminal. So let me now show you. This is the homepage of the Degronpedia. We run the query by UniProt ID type. And what we get is first the general information about the query protein, as well as the summary on the number of found degrons. Here is the link to download the results in the form of Excel. This is the interactive sequence visualizer, which shows the location of degrons on the sequence and provides not only data on degrons, but also on intrinsically disordered regions, coils, buried residues, post-translational modifications, and the pathogenic mutations. Next, there is a panel showing data on the stability of the N and C terminus of the protein. And then we move to the table section. So this is the first table provides information on all found degron motifs, as well as, for example, the ubiquitin ligases which recognize motif or any other additional relevant information, the structural context of the degron motif, for example, secondary structure, relative solvent accessibility, the disorder of the degron motif. And here, the post-translational modifications occurring within the degron motif, but also at the degron's flanking regions. And the same is for the mutation data, also within the degrons, as well as within the flanking regions. Why considering the flanking regions? Because they may modulate, for example, the degron's accessibility. And here is the section regarding the tripartite degron model. So for each found degron motif, the Degronpedia provides information of all its potential secondary and tertiary degrons. And I told you that these secondary tertiary degrons are in the neighborhood. And so this neighborhood can be defined as either in the sequence or in the structure. As shown here, there are two different tabs providing this data. Then the proteolysis simulation, either for the experimentally known cleavage sites or predicted cleavage sites. And finally, the data about E3 ligases interacting with the query protein. So the amount of output information depends on the query type. The most comprehensive is the one if you query by UniProt ID, because you have experimental data which are unavailable if you query by sequence or structure. However, if you query by sequence, you also lack naturally the structural context because you only have the data encoded in the sequence. A feature that distinguishes these two input types is the possibility to run our machine learning to predict the stability of NC terminus of the query protein. So we developed our machine learning predictors using the two global protein stability datasets from the LH lab. These data sets cover experimental stability of NC termini of nearly complete human proteome, and the performance of our final models reach approximately 0.8 R-squared coefficient. And to facilitate the interpretation of the predictions, we also provide the distributions of the corresponding stability data, and we map the predictions on them. Regarding the future perspectives, of course, it's integrating more experimental data, but also introducing new features. By new features, I mean, for example, allowing the user to screen for their degron motifs, or simulate the NC terminus streaming, which may serve as a guide, for example, in the side-directed mutagenesis, where the aim is, for example, to delete the degron site and simultaneously not create another one. And so regarding the user experience, I plan to introduce the degron visualization on the structure. I wanted to express my gratitude to Martina Bevilacqua, Natalia Guminska, and New Rawlings for their help in developing the Degronpedia, as well as all the members of the Pokrzywa lab. Thank you very much for your attention.
It's my great pleasure to introduce Tycho. Tycho Mevissen is a postdoctoral researcher fellow in Johannes Walter's laboratory at Harvard Medical School. His current research focuses on recapitulating transcription-coupled DNA repair pathways in cell-free Xenopus egg extracts. Prior to joining the Walter lab, Tycho received his PhD from the University of Cambridge, working under supervision of David Commander at the MRC Laboratory of Molecular Biology. His doctoral thesis, during his doctoral thesis, he characterized the mechanism and linkage specific of the auto family, the ubiquitinases, and developed ubiquitin chain restriction analysis, which is a powerful tool for the analysis of ubiquitin chain type, composition, and architecture.
Hello, my name is Tycho Mevissen. I'm a postdoc in Johannes Walter's lab at Harvard Medical School, and I'm very excited to tell you about an antibody-based targeted protein degradation approach called TrimAway, and how we have adopted it for use in Xenopus egg extract. In the second part, I will discuss a few new surprising insights about the mechanism of TrimAway that we learned while using this system. So the basis for TrimAway is the E3 ubiquitin ligase TRIM21, which is a cytosolic antibody adapter. And it is involved in the destruction of various pathogens after infection. And TRIM21 contains an N-terminal ring domain, a B-box, a coiled coil, and a C-terminal price-spry domain. And the coiled coil promotes homodimerization, while the price-spry domain binds to the FC portion of antibodies with very high affinity. In fact, TRIM21 is the strongest antibody receptor of the human body. So the activity of TRIM21 to promote rapid antibody-dependent proteolysis of substrate proteins has been exploited in the method TrimAway. And the current model of how all of this works is as follows. So we have a target protein of interest that is directly recognized by an antibody, which in turn is bound by TRIM21. And this is a very much simplified cartoon, but in general, the next step is TRIM21 activation and polyubiquitination. And this occurs stepwise by first N-terminal monoubiquitination that is promoted by UBE2W. And this monoubiquitin is then extended to form K63 linked polyubiquitin chains by UBE2N. And it has been proposed that these K63 chains are branched into K48 chains, but this is still under debate. So at the end of the day, this signal on TRIM21 only triggers the proteasomal degradation of all three components, TRIM21, antibody, and target protein. And this is a rather unusual mechanism because both antibody and target protein are supposed to be not directly ubiquitinated. So in the Walter lab, we use Xenopus egg extracts, which are very powerful cell-free system that recapitulates many complex cellular processes, including checkpoint signaling, DNA replication, and various DNA repair pathways. And one of the key strategies that we use to interrogate protein function is immunodepletion. So immunodepletion is basically removing or depleting a protein from egg extract by using immobilized antibodies. But this approach has various limitations. For example, you require a lot of antibody and it's very time consuming, so it's not an acute way of removing proteins with temporal control. So we were looking for an approach that would allow us to rapidly remove a protein of interest from egg extract that is untagged, and trim away was the prime candidate. So in general, you require three components for trim away. First of all, the target protein, which is using endogenous protein, antibody, and TRIM21. And all of them can be directly added to egg extract. So I started off with using full-length TRIM21 and the antibody targeting TF2S. But unfortunately, adding both components did not result in significant TF2S degradation. Fortunately, there has been a study recently to develop a minimal TRIM21 construct containing the prescribed domain directly linked to two copies of the ring domain. And this minimal construct does not require intermolecular dimerization for ring activation. So when I combined this minimal TRIM21 construct with the TF2S antibody, we saw a rapid and complete degradation of endogenous TF2S in egg extract. And this degradation is dependent on E1 activity as well as on the proteasome as expected. So we set up and established cell-free trim away in our egg extract system. So next we wanted to assess the specificity of cell-free trim away. And here we see that using a panel of different antibodies targeting different proteins, we see that only TF2S antibody degrades the TF2S protein as expected. And overall, we saw very specific and efficient trim away of different proteins, as you can see here. And there's only one feature that I would like to point out, and that is that binding proteins of targeted proteins may also be degraded, such as in the case here, where the heterodimeric UFD1 NPL4 complex is completely degraded only in the presence of an NPL4 antibody. So having established cell-free trim away, we next wondered whether we can perform functional depletion and rescue experiments using trim away. And I will show you one example for PNKP. Non-homologous end joining, or NHEJ, is one of the genome maintenance pathways that we study in the water lab. And PNKP is a kinase required for the repair of certain DNA double-stranded breaks. So we performed trim away in egg extract and saw that PNKP was efficiently depleted. And the functional assay that we use to visualize NHEJ is as follows. We start with a linear DNA molecule that is joined into these products. And when we add only TRIM21 or only antibody, we see that NHEJ activity is not impaired. However, the combination of both leads to the depletion of PNKP as well as the functional depletion, as we can see here. So importantly, adding PNKP back to endogenous levels completely rescues this defect, showing that we can indeed use cell-free trim away to study protein function. So this will be a very powerful tool in the future to use. In the second part of my talk, I will focus on some mechanistic findings that we made while using this system. Just to remind you, the current model of trim away is as follows. So we have only N-terminal TRIM21 ubiquitination that is triggering the degradation of all three components, including the unmodified antibody and target proteins. So I showed you this data before, where we looked for specificity in the system, and we saw that using different antibodies and different proteins are completely degraded in endpoints. However, this doesn't tell us anything about the fate of those proteins prior to degradation. So these cell proteins were also targeted in the time course assay. And we surprisingly saw that all of these proteins were directly polyubiquitinated prior to their degradation. So in some cases, it's a bit difficult to see, but here we see in a longer exposure that also PAX5 is completely polyubiquitinated. This is a general feature. So virtually every protein that we have successfully targeted with trim away was directly modified. So we believe that target proteins are all directly ubiquitinated. And on top of that, we would not only also detect TRIM21 polyubiquitination, but also all antibodies were directly modified. So in fact, all components are directly polyubiquitinated during trim away against them. This raises the question whether this target ubiquitination is necessary for target degradation. To test that, we used two different strategies to prevent target protein ubiquitination. First of all, we used reductive methylation, blocking all primary elements, including the N-terminus and lysine side chains. And here we see that the protein LAC-I is also efficiently polyubiquinated and degraded when we use the wild-type protein. And again, all components are ubiquitinated. And surprisingly, when we use the methylated LAC-I protein, we did not only lose the ubiquitination as expected, but we also lost all target degradation. And despite unperturbed antibody and TRIM21 ubiquitination. And so this is really not based on a lack of binding of the antibody to methylated LAC-I, because the absence of a substrate results in much lower TRIM21 activation and antibody ubiquitination. We observed the same for not only for minimal TRIM21 construct, but also for full-length TRIM21. And in general, we also observed this for another target protein, as well as with a different strategy where we mutated all lysine residues in the target protein, and that also resulted to complete stabilization. Overall, we believe that direct target ubiquitination is necessary and occurs on lysine residues because all of these lysine-less mutants are behaving the same. Next, we were wondering whether target ubiquitination is sufficient, or in other words, whether TRIM21 and or antibody ubiquitination contribute to target protein degradation. So for that, we methylated TRIM21 and antibody. And here we see that HAPA2 is efficiently ubiquitinated and degraded in the presence of unmethylated antibody and TRIM21. Now, using an antibody that was methylated in vitro results in unperturbed HAPA2 degradation, and we see even increased TRIM21 polyubiquitination. And importantly, we saw that when we prevent TRIM21 ubiquitination by methylation, this construct is still able to ubiquitinate antibody and target protein, and the target protein is still degraded by the same kinetics. And the same is true if we combine both methylated antibody and methylated TRIM21, the target protein is still degraded with unperturbed kinetics. We observed the same results also with TRIM21 filling, as well as another target protein. So in general, we show that target ubiquitination is not only necessary, but also sufficient to mediate the proteasomal degradation of the substrate. So how do these ubiquitin chains look like? In order to characterize the ubiquitin chain type composition architecture, we immobilized biotin-related target proteins on beads and performed trim away for a short period of time before removing the extract of the chains. So then we treated those immobilized and ubiquitinated target proteins with different deubiquitinating enzymes, or DUBs. And we did that in parallel reactions using the following DUBs. So we have an unspecific USP2 enzyme, a K48 specific OTP1 star, and a K63 specific induce. So this is a schematic of the approach called UbiQuest analysis, or ubiquitin chain restriction analysis, that I had developed during my PhD with David. After this treatment, we subject all these samples to Western blot analysis. We also use link-specific antibodies to get some additional information about the ubiquitin architecture. This is how the data looks like. So here we have HAPA2 again, which is efficiently polyubiquitinated on the beads, and treatment with USP2 completely collapses down this high molecular weight. Importantly, OTB1 and AMSH both trim this high molecular weight signal down. And this shows that both K48 and 63 linkages are present in this structure. Furthermore, the combination of both OTB1 and AMSH resulted in a few ubiquitin moieties being remained bound to the substrate protein, indicating that probably multiplying residues in body form. Furthermore, we can use a total ubiquitin antibody for Western blot, as shown here. And interestingly, despite the fact that OTB1 only slightly reduced this intensity of the total ubiquitin signal, we saw that K48 was completely gone in the presence of OTB. So the stub completely erased all the linkages that it's supposed to erase. Interestingly, also the K63 specific AMSH removed some of the K48 signal, as well as released some intact K48 chains. And this suggests that the majority of K48 chains is assembled on top of K63 chains. Furthermore, we detect that the K48 specific OTB1 also reduced the signal of the K63 Western blot, and also released K63 chains. And this suggests that at least a fraction of the K63 linkages is assembled on top of K48 chains. So overall, we detect that the target protein is polyubiquinated with both K48-K63 linkages, and this is a very complex structure where both chain types are on top of each other. And with that, I would like to summarize my talk. So I showed you that we have established cell-free tumor way in xenopus egg extract, which is very efficient. It is proteasome dependent as expected. And we demonstrate that this can be used to functionally deplete and rescue endogenous proteins and study their function, and we have from them form energetically. In the second part, I showed you some mechanistic insights into how tumor way works. So in contrast to current models, we see that all components are directly ubiquinated in the tumor way, that target ubiquination is necessary and sufficient. This ubiquination occurs on lysine residues and contains K40 and K63 linkages. And finally, I don't have time to show the data, but we also reconstituted in vitro the antibody-dependent target ubiquination, and the data we find there is consistent with our against data. And with that, I would like to acknowledge people in the lab, especially Johannes and Vanessa, who worked with me on this project over the last year or so. And I'm very happy to take any questions in the chat and break out the engagement. Thank you very much.